We're all familiar with the incredible life that fills our Earth's oceans, from whales and dolphins to sea turtles, fish, jellies, coral, kelp, and much, much more. But how familiar are we with the microscopic life that is ubiquitously found in oceans all over the world, and with the viruses that they have co-evolved with for millions of years? Microorganisms in our oceans are major drivers of global biogeochemical cycles, such as the carbon, nitrogen, and phosphorus cycle, all needed for photosynthesis and oxygen production. As such, they are essential backbones to marine and terrestrial food webs and ecosystems. But just as certain viruses can infect human cells, there are viruses that can infect these marine microorganisms and alter their metabolism and population density. These viruses are better known as phages, and they are found in extremely high concentrations in ocean water, with approximately 50 million phage particles per teaspoon of surface seawater. So what is the significance of these viral particles, and why should we employ efforts to understand their life cycles, distributions, and gene content? Current research estimates that marine viruses are responsible for the mortality of 1/3 of microorganisms in the water column per day, which drastically decreases microbial diversity and abundance in our oceans. Killing of cells by phages is referred to as a virolytic cycle, where phages attach to their specific host organism, eject their DNA into the cell, and then hijack host cell machinery to replicate DNA and viral particles. Once phage particles are assembled, the cell membrane is lysed and phage particles are released, ready to infect other nearby hosts. An estimated 140 gigatons of carbon are released from these lysed cells every year in a process termed viral shunt. In this way, carbon in the ocean is prevented from reaching higher trophic levels and is instead shunted to lower trophic levels as dissolved organic carbon. However, this isn't the only life cycle phages can carry out. Temperate phages can also undergo the lysogenic cycle, in which they inject their DNA into their hosts and integrate it into the host genome without killing the cell. By some estimates, 20 to 40% of all aquatic bacteria are infected by phage DNA and are thus known as a virus cell or a prophage. What's significant about this is that viral DNA integration into the host genome can lead to metabolic reprogramming of host cells. These viral genes are collectively referred to as auxiliary metabolic genes, or AMGs, and they can have a profound effect on biogeochemical cycling of elements. Viral particles generally acquire AMGs from DNA recombination within an infected host cell. They are advantageous to the phage because these genes can allow for the production of extra energy through host metabolic processes, such as photosynthesis, that can then be funneled into phage DNA replication and particle assembly. AMGs can also function to shut off certain processes altogether and redirect carbon and other resources to phage replication. Given the fact that phages are able to pick up DNA from their host, these viruses display the most extensive genetic diversity in the entire ocean. With advancements in metagenomic sequencing in recent years, viral particle distributions are starting to be studied and understood. However, more work needs to be done to elucidate the effects of viral infection on host populations and better incorporate viral species into global biogeochemical models, especially given the context of our current climate crisis that is heavily impacted by atmospheric carbon export occurring in our oceans, which can be altered by viral shunt and the metabolic reprogramming of host microorganisms infected by phages.